When you picture scientific research, you might imagine scientists studying thousands of completely different organisms for every different question. And technically, they could. There are millions of species on Earth, all with unique biology. But in practice, scientists tend to come back to the same small group of organisms again and again. These are called model organisms. Today, we're jumping into what these model organisms are, why we work with them, and how we work with them. A model organism is basically a species that scientists know really well. Its genetics, how it grows, how it reproduces, even how it responds to stress. Because researchers around the world have spent decades building knowledge and tools around these organisms, they become really efficient systems for asking scientific questions. The same organism can help answer totally different questions. One lab might use it to study cancer, another to engineer biofuels, another to understand evolution, and another to build biosensors. Instead of starting from scratch every time, scientists build on this giant shared foundation of knowledge. That's especially true in iGEM, where teams often need organisms that are safe, predictable, and easy to genetically modify. So even though our projects are wildly creative, many of them begin with the same familiar biological toolkits. What are these magical organisms that everyone uses then? The most common one is Escherichia coli, or E. coli. I know, some of you might have only heard bad things about E. coli. We really don't like E. coli in our food, but we do like it in the lab. The numbers behind that are pretty crazy. E. coli appears in more than 300,000 scientific papers, making up roughly 21% of all publications focused on bacteria. So what are the genetic tools and methods that we spent decades on? The most important of them is the transformation protocols. Here's Gloria and Yen to tell us about heat shock transformations.
The first step of transformation is called incubating on ice. So what we do is we literally just get a solution with our plasmid. We put it into a tube of competent cells for E. coli. So what are competent cells? They're like E. coli cells that have been frozen and they have thinner membranes than usual, so it's easier for us to introduce new DNA to the cells. We put the solution into E. coli and then we let them sit for about half an hour. And the reason for that is we want the DNA to sort of bind around the membrane of the cells, just on the outside, because DNA is negatively charged and also the nuclear membrane of the cells that we use are also negatively charged. So they usually repel each other. So we want to give them some time to sort of get used to each other. Sometimes we add a chlorine solution, I believe it is, that also helps to neutralize the charge. But then after the 30 minutes of incubation on ice, then what we do is heat shock. So we put the cells in a bath of warm water. It's like usually 40 degrees Celsius. And the cells are very sensitive to heat. We usually keep them at like about negative 80 degrees Celsius. That's like the perfect temperature for them to not freeze. If you put them in 40 degrees Celsius, the cell membranes basically become really, really thin. And that's when you can flush the DNA into the cells and through the membranes. So we do heat shock about 30 seconds. We don't want to do it for too long because then the cells will die, because being under 40 degrees Celsius is very, very stressful to them. So then after you do that, you put them in a tub of ice again to let them cool down because they're really, really stressed out again. You want to give the membrane some time to heal back together. And then after that, you can put the cells into a shaker, and the shaker usually is about 37 degrees Celsius, which is the optimal temperature for the cells to grow. You put them in the shaker and the shaker shakes the cells as they're growing, and that just helps the cells grow a lot faster. And then you have a culture with a bunch of cells, and then you just plate the cells onto antibiotic plates. Another thing to mention is that the plasmids usually have some kind of antibiotic resistant gene, and the reason for that is we want to select for colonies that only have the plasmid that we want. And E. coli is usually not resistant to a lot of antibiotics. For example, like I use ampicillin. E. coli is usually not resistant to ampicillin. So the only way for E. coli cells to survive on a plate with ampicillin is if they got the extra DNA from our plasmid, which has the ampicillin resistant gene. That's how we know which cells have potentially the plasmid that we want. So that's like the basic process of transformation. So obviously not every iGEM team will use the same organism, but the process of transformation, cloning, everything is relatively simple. The biggest difference is that probably depending on the organism and what you're trying to use it for, there would be different efficiency rates, especially if you're trying to use a non-model organism.
We may not want to use E. coli all the time. It is a pretty great model organism, but it's not perfect. Let's talk a bit more about situations where we don't use E. coli.
E. coli is technically like a foodborne illness. It could be harmful for you, especially if we're doing something that people will touch. Our project this year, it involves direct contact with human skin, and it could be a concern for others, like putting E. coli onto your skin, because E. coli does give you food problems and stomach problems. Another reason that we found was secretion problems. E. coli tends to keep proteins inside the cell rather than exporting them efficiently, and we need a bacteria that will properly excrete the protein we need.
As an iGEM team, we do have some limitations in what we work with. Of course, we want everyone to be extremely careful with what they're genetically modifying. iGEM has guidelines and approved organism lists designed to keep projects safe for students, researchers, and the environment. Most of the organisms commonly used by iGEM teams are considered low risk and well understood. The reason these organisms are favored is practical. Scientists already know how they behave, how to contain them safely, and what kind of risks they do or don't pose. As I mentioned before, they're also organisms with lots of existing genetic tools and published protocols, which makes projects more accessible for student teams. Let's talk about two of these organisms. First, Bacillus subtilis. Many bacteria require artificial methods to make transformation possible. B. subtilis, however, has natural competence. When environmental conditions are right, such as nutrient limitation and high cell density, the bacterium actively searches for and takes up DNA from its surroundings. Specialized proteins on the cell surface bind to pieces of DNA in the environment. One strand of the DNA is broken down, while the other strand is transported across the cell membrane and into the cell's interior. Once inside, the imported DNA can pair with matching sequences in the bacterium's chromosome through a process called homologous recombination. One of its most important abilities is sporulation. When conditions become unfavorable, whether that be low nutrients or extreme temperature, B. subtilis can transform itself into a dormant structure called an endospore. These spores are incredibly resilient, capable of surviving extreme heat, dehydration, and other environmental stresses that would kill most cells. Because of this, researchers often use it to study how cells respond to stress and make developmental decisions. It also naturally secretes proteins into its environment, making it a good choice over E. coli sometimes, as Gloria mentioned. This means that when scientists engineer it to produce useful enzymes or other biological products, those products can be collected much more easily. Another commonly used organism in synthetic biology is Lactobacillus plantarum. L. plantarum is a species of lactic acid bacteria found naturally in fermented foods, plant material, and the human gastrointestinal tract, which are environments that scientists may want to engineer directly. However, transforming it is significantly more challenging than transforming B. subtilis. Unlike B. subtilis, it does not actively take up DNA from its environment, so researchers use other artificial methods. The most common method is electroporation. In electroporation, you're essentially just shocking cells with electricity to open up tiny holes or pores in their membranes. This lets you sneak in DNA, RNA, or proteins. However, you have to make sure to watch your salt. Salt conducts electricity. Too much salt causes an arc, which is basically a mini lightning bolt inside your tube that fries your cells. Nobody wants exploded cells, so step 1 is preparation. You have to wash your cells thoroughly to get rid of growth media salts, and then suspend them in a low salt buffer. You have to make sure to keep all of this on ice. Step 2 is the mix. Add your DNA directly to your chilled cells, but keep the DNA volume tiny, usually just a couple of microliters, so you don't introduce salt. Step 3 is the pulse itself. Lock the cuvette into the electroporator and dial in your parameters. You're going to have to know the voltage and the specific kind of pulse for your specific cell. Then you let the machine do its thing. Finally, step 4 is recovery. The cell membranes are damaged and leaking, so you have to act fast. Within seconds of the pulse, you have to flood the cuvette with warm, nutrient-rich recovery media. Similar to heat shock, you put them back in the incubator so that they can heal their walls and start expressing their new genes. Even with electroporation, transformation efficiencies are often much lower than in organisms such as E. coli. Lactobacillus species possess thick gram-positive walls that can physically hinder DNA uptake. B. subtilis is also gram-positive, but it's much more capable of taking up DNA because of the reasons we mentioned earlier. In addition, many strains contain restriction modification systems, which function as a bacterial immune system. These systems recognize foreign DNA and cut it apart before it can become established inside the cell. As a result, researchers often need to carefully prepare DNA, modify growth conditions, or use specialized plasmids designed specifically for Lactobacillus species. Now, what about model organisms that aren't bacteria? We can also work with things like fungi and plants. Here's Christian Funkelrud to tell us about what it's like working with fungi and plants.
Fungal and plant transformations are really cool and they're super important to biotechnology. So I previously worked with the plant model organism, Arabidopsis, and to genetically modify these plants, we use Agrobacterium mediated transformation. This uses the bacteria Agrobacterium tumefaciens, which is a natural plant pathogen. Its ecological niche is in the soil and rhizosphere, but it can also be opportunistic as a plant pathogen. Essentially, when the plants get damaged, whether it be through insects eating them or through mechanical damage, the plants start secreting hormones to signal that they're damaged. One hormone that the plants make is called acetylsyringone, and Agro can recognize these hormones and it'll act like an invitation to them. The bacterium activates this special genetic system where they can attach to the plant cells and actually modify their DNA. The bacteria makes a single-stranded copy of tDNA, which is tumor-inducing DNA. It attaches this DNA to protective proteins, forming a T complex. The bacteria then builds a protein bridge that inserts the T complex into plant cells. This moves through the nuclear pores of the plant, and the DNA randomly integrates itself in the plant's own genome. The bacteria doesn't infect in the traditional sense because it's actually changing the DNA of plant cells. This sequence causes the plants to produce excess growth hormones, which leads to crown gall tumors. You've probably seen these on trees before, where there's a lump on the outside of the wood. The bacteria eat the excess sugar that the plant is producing and grow in the tumor. In biotechnology, we utilize the fact that Agro can edit plant genomes. We can modify the T plasmid, which is the one where tDNA originally is from, and insert a gene we want into the plant instead of the tumor-inducing gene. In the lab, we can replicate our recombinant plasmid in E. coli, and then we electroporate it into Agro, and then finally we can insert this recombinant DNA into the plant. The actual process of mediating this transformation in the lab is growing a ton of engineered Agro, resuspending it in a solution of sucrose and a surfactant, and then dipping mature flowers into this solution. This is called floral dipping. We wait for these plants to dry out and die, then we can harvest the seeds, and then in the next generation, we select for plants that were correctly edited by Agro because we added an herbicide resistance cassette when we do these transformations. In the lab, we use this to make CRISPR knockouts of certain genes of interest. It can also be used to modify plants that have insect resistance or to improve nutritional content. Now I work with two fungi, a yeast and a filamentous fungi. We actually use the same transformation with the yeast. It's called Rhodotorella torulloides, and it's a rising basidiomycete yeast model which can be used industrially for biofuels as it can accumulate up to 70% of its biomass as lipids. We use Agro to modify this yeast too, except it doesn't produce the right hormones to induce Agro to do the transformation because it's a fungi, not a plant. So we add the hormone acetylsyringone to the induction media when doing the transformation, and we mix the Agro and yeast together and then grow them on selection plates with a lot of antibiotic to kill the Agro and with the specific drug that we gave the yeast resistance to. Another way to transform yeast is with lithium acetate transformations. You'll commonly see this in Saccharomyces cerevisiae, which is the model Ascomycete yeast. So fungi have tough cell walls that are negatively charged. DNA is also negatively charged, so it can be hard to get the DNA inside of the cell. First, lithium cations neutralize the cell wall and soften it. Polyethylene glycol, which is a crowding agent, is then added and pushes the DNA against the cells. The cells are then heat shocked to disrupt the cell membrane, allowing DNA to flow into the cell, and the yeasts are then grown on selection media, and now you have genetically modified yeast. Yeast is a very important organism in biology because it is a eukaryote and it is relatively easy to work with. Much of our fundamental knowledge of cell biology comes from studies originally done in Saccharomyces cerevisiae, and on top of that, oleaginous yeast like our torulloides, the one I'm currently working with, have potential applications in industry for renewable biofuels or transitioning to the bioproduction of other compounds that are traditionally made from petroleum. Finally, I wanted to talk a little bit about filamentous fungi transformations. So I also work with a mold called Neurospora crassa. It's the primary model filamentous fungi, and we also use electroporation on it. Neurospora produces asexual spores called conidia, and generally when they're germinating, they have weaker cell walls. We collect these conidia in a sorbitol media and filter them through a cheesecloth to filter out mycelia or other debris. We wash the conidia and then electroporate them with our target DNA, and the cell does the rest. Neurospora is a really cool organism to work with because there's been a lot of fundamental research in it on circadian rhythm, gene regulation, and metabolism. George Beadle, who's actually a Cornell alumni, won the Nobel Prize with Edward Tatum for the one gene, one enzyme hypothesis, and they used Neurospora as their model. There's also a lot of really cool research on using filamentous fungi as biomaterials.
To conclude with this episode, let's expand outside of iGEM and talk about an application to model organisms which are used in other kinds of labs.
The model organism for flies is Drosophila melanogaster, but actually what I think is really cool about my project is that I don't just work with melanogaster. I work with a bunch of different Drosophila species, like Drosophila melanogaster obviously, and also Drosophila simulans, and Drosophila jacoba. So those two lines, they're sister species of melanogaster. They're pretty similar, but they do have a lot of unique differences, and that's what I was researching in my lab, like the little different protein differences between related species. The reason why Drosophila in general is considered a model organism is because it has very short generation time. Like, it takes 10 days for a Drosophila embryo to become a full adult. They also have pretty relatively simple DNA structure. They have 4 chromosomes. One of them is the sex chromosome, and one of them is so small that most people don't even care about it. So really, you're just working with 3 different chromosomes. The way these genes work is sort of similar to how it works in humans. So whatever we find in Drosophila DNA is often applicable to human-related issues. When we do CRISPR edits, which is when we make a specific edit in a species, we need a vector of some sort. We can't just directly make a change to the gene. And the way we usually do it is we use E. coli as the vector. So we add whatever genetic annotation that we want into the E. coli. And then we use the E. coli to make the DNA, and then we inject that DNA into our flies or onto whatever other organism you want. I just said flies because I usually work with flies. In my lab, I use E. coli as a vector to make the DNA I want, and then after I get the DNA I want, we send them to a company that takes the DNA and injects them into the embryos of flies. So then when the flies grow up, they have that DNA mutation in them. One thing to note that I thought was very interesting is we inject the embryos of the flies, right? If you know anything about development, you start with like a single cell, like a zygote, and then that cell becomes 2 cells and then 4 cells, and everything divides. Obviously, when we inject the DNA into our embryo, we're not going to inject it into the zygote, like the very, very first cell. It's just too small, like we can't physically do it. Some of the cells are the wild type cells, which is just like the regular fly DNA. But then some of the cells that we injected into, so we inject into one cell and that cell divides. So the descendants of those cells will have the mutation that we want. So we end up with what's called a mosaic organism, which is that some of the cells have the mutation, some of them don't. So we can't actually use that cell, or have to use that fly and then cross it together with its own siblings. And then after that, you can get a full fly with just the mutation you want. Because if you think about it, every child of a fly comes from a single cell from that fly. So if we use that cell that has the DNA mutation, then whatever descendants follow will have that DNA mutation. So I just thought it was something very cool and not intuitive, because usually we're like, oh yeah, it's like hand wavy, like, oh, you inject DNA into the embryo and then something happens, but it's usually a lot more complicated than that.
This marks the end of our iGEM biology crash course. I hope you all enjoyed it, and hopefully you'll be able to use your knowledge to impress a professor, your friends, or iGEM judges. I encourage everyone to look more into synthetic biology, because the possibilities really are endless. Thank you all for listening to this series, and make sure you stay tuned for our next series, Science for All, where we talk with our Wiki and Design team about making science more accessible. Until then, stay smart, and I'll see you geniuses soon.